Hello, everybody. This is Mrs. Pound, and I am here for the last video of Chapter 1 um, on the Science of Life and the God of Life from BJU Biology 4th edition. And today's video is going to be from Section 1C, Biology and the Study of Life. So our objectives today are going to be to list and describe the attributes of life and to identify the parts of a light microscope. So let's first begin by talking about what biology is. Biology is the science that deals with living organisms and vital life processes. So a good way to sum that up is to say that it is the study of life. Now, as we look at the attributes of living things, we're going to discover that non-living things may possess several of the attributes of life. So it can actually be kind of tricky determining whether something is living or not. And we're going to take a look at that in class and do an activity on that. So let's talk about first the attributes of life. So in order for something to be living, we say that it has the living condition. This is a highly organized cellular condition that is derived from pre-existing life and that faces death, requires energy to carry on processes such as growth, movement, reproduction, and response. So a lot of the attributes of life are packed into this definition here. So let's unpack this and take a look at each of these things. First, life exhibits movement. And what I'm talking about is mostly internal movement. This is an attribute of life that includes the movement of fluids, organs, and even structures within cells. So there is internal movement inside of cells, and I have blood here. There's movement of blood through us. Now, this does not mean to move from place to place. That is locomotion. And many living things do not exhibit locomotion. You know, plants do not tend to move from place to place. Um, there are even animals that do not move from place to place. Things like a lot of clams tend to be very fixed. Cnidarians, some of them tend to be very fixed in their locations. I'm thinking especially of Hydra. They do not move around. They stick to rocks, okay, and stay in place. They achieve growth. And a process involved in this is called assimilation. This is the conversion of nutrients into living cells, and it is a process of growth. In order for a living thing to grow, it needs nutrients. It needs nutrients. And there are other processes that we're going to talk about in the attributes of living things that help them to acquire these nutrients. And assimilation is when an organism organizes simple molecules into complex living materials. So they take in simple molecules and make more complex things out of them. Living things also reproduce. A reproduction is a formation of another organism that has characteristics and limitations similar to the original. So here we have a koala bear, and there is a little koala bear on its back. And so with the reproduction, what it means here is as far as another organism similar to the original, you know, a koala bear is not going to give birth to a tiger, right? Because that's an animal of a different kind. So that's what we mean by that. It comes from similar pre-existing life. This is biogenesis, the principle that living things only come from living things. It's not spontaneous generation. They cannot come from non-living materials. And again, you know, you're going to get a hippo from a hippo, um, not a blue whale, okay? They also have variations. This is the difference between individual organisms of the same kind. Differences based on genotype, the expression of different individual characteristics of the same kind. So it's in the genes that these variations are. And we have variations with a type of reproduction called sexual reproduction. They also have similar chemical makeup. Living things tend to be made up of organic compounds. These are naturally derived from living organisms. Organic compounds are those that contain carbon and also hydrogen. Okay, so in the back, we tend to use organic to mean foods to be that they're not grown with pesticides, but that's not the case here. Organic means that it contains carbon and hydrogen compounds. Also, there are inorganic compounds. This is describing objects that are not alive and that have never been alive. Also, substances that lack carbon, with few exceptions. Carbon dioxide is an exception because it's just carbon and oxygen, no hydrogen there, and usually derived from non living material. Like this watch here is made of metal, not carbon. Now, living organisms are composed of cells. We're going to study cells extensively, so I'm not really going to go into that here, but that is a characteristic of life. They also exhibit irritability. So that doesn't mean that they just get angry. Irritability is the ability to respond to changes in the environment. So we see a deer here in the background with its ears perked up and looking around. It probably heard something. It is responding to the environment. So it's showing irritability. Living things also require energy. Now, we obtain most of that energy from our food. This is organic energy containing substances required by all living things. Now, plants do not require food, but they make it. And actually, they require the food that they make. They cannot just take the energy right from the sun and utilize that energy. They have to make it into food first before even they can use the energy. So, all living things do require food. They also maintain a high level of organization. Um, As far as high level organization, there are different levels of organization. There's organization at the cellular level, but even a cell is highly organized. It's not just an empty box as cells were thought to be at first. They are highly organized, containing a lot of parts that have to work together. Cells can be formed in higher organisms into tissues. Tissues can be organs, and organs, organ systems, and then finally an organism. Now, different organisms can have different levels. Some things, like a sponge, only go to the tissue level. Okay, so it depends upon the organism and how complex they are. All living things also face death. They will die at some point. There are different lifespans, though. Some organisms live for very short periods of time. They may live for a day or less. Other things, have found trees that have been alive for 1,000 years or longer. And so just life is the living condition. Okay, so that's a simple definition for life. Now, how do we study life? One thing that we will commonly use in this class to study life are microscopes. This is a scientific instrument that magnifies objects for more detailed study, usually contains a series of lenses. And we'll talk about those lenses in class. Now, the father of microscopy is Leeuwenhoek. Okay, he was Dutch, and it was actually a side hobby of his. He was not a scientist per se, that was not his day job, but it was a hobby, and he really came up with the first microscopes. These are some drawings in the back of his first microscopes. Look nothing like ours today. Today, we use a compound light microscope in our class. And the compound light microscope, some of the important parts are the objectives. This is the part of a light microscope that is near the specimen and that contains lenses which form an image of an object. The objective lenses, remember, are right here on that revolving nose piece at the bottom here. They're on the revolving nose piece, so magnification can be varied. So those are those objectives. There's also the ocular. This is the eyepiece of a microscope, and it contains lenses as well. So here's the ocular that you look into. There is a stage, the flat surface of a microscope on which a slide or specimen is placed. So right here, the stage, and the body tube. This is the cylindrical part of the microscope between the eyepiece and the objectives. Now let's talk about the principles of light magnification to help us understand how microscopes work. First of all, there's reflection. This is the image caused by light rays bouncing off of an object. This is the reason we see things, is because there has to be reflection of some kind um, in order for us to see objects. There's also refraction. This is the bending of light ray when it passes from one medium to another at an oblique angle. And what we do is the lenses are curved, and that curvature of the lenses causes the light to bend, and we can magnify objects by bending that light. Also involved in magnification is resolution. This is the characteristic that allows a microscope to form a clear image of detailed structures. Different microscopes have different resolutions depending upon the types of lenses they have in order to form images. Some have better resolution than others because some have better optics than others, have better lenses. Um, and so all of that is dependent. And they all have a resolution limit where they just cannot have any better resolution. That is all they are going to magnify clearly. We also have techniques for modern imaging. These are not techniques that we use in our laboratory, but they are things that maybe you have encountered. The first is the electron microscope. With an electron microscope, we can see things like viruses. This is a high-powered magnification device that uses streams of electrons rather than light waves to form an image. It's a tool that makes an enlarged image of an object's surface, and it uses a screen instead of an eyepiece. So this is very involved, and it uses computers in order to get those images. Now, an electron microscope can see things that are very small. Uh, with our microscopes in class, we're going to see things that are maybe millimeters small. An electron microscope can go down to nanometers. Now, if we take a look at that, a meter contains 100 centimeters, centi like century, 100 years. A meter contains 1,000 millimeters, milli meaning 1,000, like millennium, 1,000 years in a millennium. Micro actually means 1 million. So there are 1 million micrometers in a meter. And nano means a billion. There are a billion nanometers in a meter. So micrometers and nanometers, if I had a meter stick with that fine of lines on it, it would just look like a black strip because these are such small distances and such small things that are being measured. Also, there are MRIs. This is an imaging technique that uses magnets. This is why when you go in for an MRI, you cannot have metal on because it will be attracted to those strong magnets and it would hurt you. Also, you're asked if you have any internal metal because like any screws or anything from surgery because that could cause damage or shift. So you cannot have metal that would be attracted to those magnets. Now let's take a look at some variables in biological studies. First, we have to identify biological variables, which can be difficult because it's difficult to control all of the variables in a biological experiment. For example, if you're doing an experiment to identify the effects of exercise on an individual's heart health, uh, there are other things that are difficult that also affect heart health that are difficult to control because maybe a person's diet, you're unable to control that, or their genetics. Maybe they're just prone to certain diseases or things. So we cannot control everything in biological studies. Also, we have to take a look at the validity of biological studies. There can be errors made in observations. For example, I like to use the example of two men observing something. One is looking through a hole in a fence, the other is looking over the fence. The man looking through the hole says, "Oh, it's raining out." The man looking over the fence says, "No, a person is watering their yard with a hose." Um, it depends upon your perspective what kind of observation you're making and how you're making that observation. It could lead to errors. Errors can result if the specimen is atypical. We have to make sure that we're looking at a healthy specimen that does not have any, that's like the rest. Maybe we have a diseased specimen and we're not going to get good data. Also, errors can be made when the techniques used are crude. We have much better microscopes now for making observations compared to what they had previously. And so, the better our technology, the better our instruments, the better our data is going to be. Okay, so our objectives for today's video were to list and describe the attributes of life and to identify the parts of a light microscope. Now, don't forget to continue playing this video until you answer the questions at the end. And also, don't forget your 5 questions in your notes. Also, this is the last video for this chapter. So you can start studying Quizlet, you can start studying for your test. There are also questions at the end of this study guide to help you study for that test.